I tell you, I'm so pleased today that I'm going to get to introduce Lisa Ball, who is a member of our church. It's been since 2010. I just read her name tags. And her husband, Mike, where'd you go? Oh, you're Mike Gross. So both of these two great biologists and longtime friends. Lisa grew up in Texas. She got smart and came to Kansas to do her graduate studies. And so at KU, she was studying the tropical bird conservation in Costa Rica and Mexico. And that's where she met Mike, as he was studying fish evolution. She worked briefly in environmental consulting, but was pulled back into her first love, education, as an adjunct at KU and Highland Community College, before going full-time teacher with the Lawrence Public Schools. This is her 19th year of teaching secondary science, and she is currently teaching environmental science at Lawrence High. Lisa continues to bring real-world science into her classroom as often as possible. She was a member of the Toyota International Teacher Program in the Galapagos Islands in 2010, an Earthwatch Teacher Fellow in 2009 and 2017. She received the honor of the 2020 Keep Kanza Teacher of the Year, and in 2021 she had the joy of sailing the Pacific as a Science Communication Fellow with the Ocean Exploration Trust on the ship EV Nautilus. In August, Lisa was awarded a Fulbright Teacher for Global Classroom Award by the U.S. Department of State, which means she's trained to incorporate global competency into her classroom. She will be part of a global symposium in Washington, D.C. in February, and she will be part of an international school exchange somewhere in the world this coming spring or summer. When she's not teaching or sciencing, she loves to spend time gravel biking, and she and Mike have been converting their yard into a native pollinator garden. They love to hang out with their son Logan, and we watch Logan grow up here in the church. We all love Logan, who is now a senior in college, and he's studying wildlife biology. I believe he's in Nebraska, and he's done some exciting things too. And Dad suggested maybe we could have him come give us a program too of what he's been doing. So Lisa, I'll let you
All right, thank you so much. I am so excited to be here, and I appreciate the invitation to share. As I was telling Jan, it's always nice to talk to an audience who actually chose to be here, like my students. And just to follow up, I just got my international placement over the holiday break, and I will be traveling to the country of Ghana in West Africa in March. So I'll be there for a few weeks, visiting schools and learning more about global issues and about environmental issues in their country and education in their system, and making partnerships back with the United States. But now I'm going to talk about the ocean, so I thought I'll put on my sailing vest and go on a little journey. So this was a real treat for me to be a part of this program, and I was very honored to work with amazing people. These are all people who were selected for the position that I had during the 2021 year. And I'll tell you a little bit more about how this all came about. So as you all know, we are about 3.5 hours from the geographic center of the United States, right? And so because of that, many of my students at Lawrence High School know very little about the ocean. Many of them have never even seen the ocean. And so I'm always looking for ways to bring that information back into my classroom, because as you know, or may not know, the ocean covers about 70% of our planet. So not knowing about 70% of your planet is a real problem. So the title of my talk has two meanings. As I mentioned, many of my kids don't know much about the ocean, so I'm bringing it into my classroom, but also just as a reminder that 85 million years ago, we were sitting in an area that was a shallow ocean. And, you know, we still have the remnants of that ocean left behind, our beautiful chalk formations that cover western Kansas. This is Logan, by the way. This picture was taken at Little Jerusalem in 2020. Beautiful formation if you haven't gotten to visit it. I believe it's still our newest state park in Kansas. And these beautiful limestone formations were formed from these microscopic sea creatures that lived in our shallow ocean. And this is an example of one that was very common at that time. And when they died, their little skeletons fell to the bottom of the sea and were compressed and made the limestone that we live on. So kind of thinking about it, really, we've got the rock chalk jayhawk shanty. Think about it, it's almost a sea shanty. So there's our ocean connection for Kansas. It's actually one of our original biomes. All right, so as a new teacher in 2003, I was invited to go to a teacher conference, science teacher conference in Kansas City, and I ended up in a session with a presentation by this man, Dr. Bob Ballard, and got really excited about his tales of ocean exploration. Found out he was actually a Kansan. He's from Wichita. He was born in Wichita. And as a little boy, he had dreams of traveling to the ocean, exploring the ocean, but he lived in Wichita, Kansas, so, dreams can die there, right? But he kept his dream. He also struggled in school with dyslexia and ADHD. And, he was the kind of kid that a lot of teachers might have given up on, but he kept on. And his family ended up moving to California, and he became a pioneer of ocean exploration. Happy ending of the story, he has multiple PhDs and 22 honorary degrees. He's known for the type wreckage, as you may know, and documenting that story with the ocean technology that he helped to pioneer and design. And he's also been credited with finding many other shipwrecks as well. However, he would like you to probably know him best for this finding. When I was just 6 years old, 1977, he was also one of the first scientists to come across formations such as these, doing deep sea exploration. These are the hydrothermal vents. Because he was with a team of geologists and not biologists, they could only guess at what they were seeing. They had no idea that there would be life that far down in the ocean. But they took samples, so they smelled awful. And they brought them back, and with the help of biologists, they figured out that these indeed were the tube worms that you see up here, giant tube worms that are filled with symbiotic bacteria. And they live solely off of the inorganic materials that are coming out of the crust of the ocean, things like methanes and sulfides that we would have thought would be toxic to life, but indeed are supporting another form of life. More recently, he's gotten some press for his search for Amelia Earhart's wreckage. And so this is a really cool show if you have access to it. I know it's on Amazon Prime and Disney Plus, Expedition Amelia. And it's the story of them looking for the airship. Spoiler, they didn't find it, but they have some really good ideas and they're hoping to go out on another expedition. So 2 more Kansas connections. She was, of course, from Atchison, Kansas. If you haven't been to the Amelia Earhart Museum in Atchison, it's delightful. I recommend it. And then also, this is Allison Fundus, who was the leader of this expedition. And the reason why, a little precursor to the story I'm coming up on, one of the reasons why I got involved is because her cousin is Julie Boyle, who some of you may know through Lawrence Public Schools. So she helped us get connected. So in 2008, Dr. Ballard founded the nonprofit organization that he called Ocean Exploration Trust, and he acquired several vehicles for his exploration. Here you can see the ship that he purchased, which is the exploration vessel Nautilus, and I'll talk more about it and its history in an upcoming slide. And then these 2 exploration vehicles, which are called remotely operated vehicles, or ROVs. This little one over on the side was named Argus, and the one on the right is Hercules. And the goal of his program, the aim of OET, is to explore the ocean, seek out new discoveries in the field of ocean science while pushing the boundaries of education, outreach, and technological innovation. So back to my personal story. Fast forward to 2019, and I saw a late night email from Julie Boyle in my inbox. Probably the other teachers were sleeping, so it worked to my benefit. And she said, Dr. Ballard is coming through Lawrence. He was coming to visit some family in Kansas. And would we like him to speak at any of our schools? And of course, I jumped on that right away. And we had a great day him presenting to our students. And once my students kind of figured out who he was, they were pretty starstruck. He's a really great, charismatic speaker. And before he left that day, his team asked me if I would be interested in going and exploring the ocean with them. Of course, I said yes. So I applied and I was accepted into their teacher program. But of course, you know what happened in 2020. So we all got tabled. And then 2021 rolled around and they thought, well, this is still not going to happen. And then all of a sudden in the fall, I got a kind of a last minute email that said, hey, we're going to try this again. And so I was invited to travel with them in December of 2021. So in December of 2021, I flew to Honolulu, Hawaii, and joined the crew of the Nautilus and port there for our journey. And this was only my second time on a ship and my first time in Hawaii. I was very excited. And it was extra special because Dr. Ballard doesn't travel much anymore, but he was on the ship with us for this expedition. He's now 79 and still, sails like a pro. And he had just published his new autobiography, Into the Deep, which I have a copy of if anyone's interested in looking at. It's a great read. He had written a previous autobiography, but they were able to update it because some of the information from his Navy days has now been declassified. So it has some of those secrets in there. I'll read them. He's a nice guy. So this is the core of exploration. It's a team of 33 scientists who are working together on the ship for this expedition. The ship can hold 50, so the other people were the crew. We left port from Honolulu and traveled for 24 hours to explore for 12 days. And we only saw one ship the whole time we were out once we got away from port. So we were in a pretty remote location. So the name of my expedition was the Chautauqua Seamounts. But as soon as we got on the ship and we had our first meeting, they said, we never should have named it this. Don't call it that. Because in fact, a seamount is just an underground mountain. And only one of the mountains, seamounts, is named the Chautauqua, and the rest have no names. They're still unnamed. So we know so little about them, they have not even been named. But that was kind of the best they could do. They wanted to make sure that we understood that most of the chain of these seamounts, they don't even have a name. So we were going to be mapping them and exploring them, trying to find out more about their origin, and collecting samples with the ROVs. So two main goals of the expedition: to understand and document the biology, which is the side that I was the most interested in, but also we had geologists with us who were very interested in sampling the iron manganese rocks at the bottom of the ocean to find out more about how these seamounts came about, the age and the origin of the chain. They had some hypotheses they wanted to test, and also to look for rare metals such as cobalt. And you can see the scientist here is looking at and measuring some of these rock samples. And this is just an example of what this deep sea life looks like. We call these deep sea sponge and coral gardens. And they look very different than what you think of when you think of tropical coral. Most people think of Hawaii and the Great Barrier Reef and these brightly colored organisms. But this is at the bottom of the ocean, keep in mind, where there's no light, no photosynthesis. The temperature's near freezing. So think about living in conditions like that. It's a very different ecosystem. And so the sole source of food for these creatures living down here is called marine snow. And marine snow is basically poop and dead stuff falling down from the surface to the bottom of the ocean. And then also little zooplankton. And so growth is very slow with these conditions, and many of these organisms can't live anywhere else. So again, we were in the deepest part of the ocean, 4,000 meters 13,000 feet down, about 2.5 miles down for the exploration. At least that's where the exploration was. We stayed on the ship, I'll clarify that in just a minute. But our exploration was 2.5 miles below the ocean in an area where there's no light, the midnight zone. So the biologists thought that perhaps we would find the highest biodiversity on the slopes of these seamounts because the current is washing more of the nutrients up to that area. And so that was kind of the working hypothesis. We didn't know what we would find, but that was kind of their idea, is that we would be starting typically at the bottom of the ocean and exploring from the base of the seamounts all the way up toward the top to document what was there. So back to the ship. The Nautilus is one of the few ships in the world that's devoted entirely to scientific exploration. And it was purchased as it was an East German fishing exploration vessel, and it was retrofitted for deep sea work. It's 224 feet long, it has 17 permanent crew, and it can house up to 33 additional members, like I mentioned, who are there for scientific exploration. And it's explored the ocean around the world, but right now the focus is in the Pacific. So in 2013, the Corps added a new hull-mounted multi-beam sonar system, and this system can efficiently map the seafloor in waters from 10 meters to 7,000 meters deep while the ship cruises along. And let me tell you, it's moving really quickly when it's doing this. I was quite surprised. The sonar collects bathymetric data, so the texture of the ocean, surface sediment characteristics, and water column data. And this information helps them identify areas of interest to go back and explore with the ROVs, and just creates maps that have never been made for this part of our planet. And you can see what these images might look like in the inset photo. This is a 3D image that was generated from this mapping. And you probably, most people think the bottom of the ocean is just flat, but it's not. It has mountains and plains just like we have on the surface above the water. And again, even though the ocean covers 70% of the planet, we know more about the moon than we know about this part of the ocean. So there's a great deal of work to be done. In the earliest days of ocean exploration, scientists like Dr. Ballard risked their lives going down in deep sea diving vessels. They'd spend more time traveling down than they would be able to spend at the bottom of the ocean. It was a real treat that he was there with us and was able to tell us some of those tales of near death and sleeping in tiny hammocks lined up one over the other in these vessels out in the ocean. And so he had a dream of making this safer and easier, so he helped with the innovation of these new vehicles that we call ROVs. So these remotely operated vehicles are basically robots that do that work for us now. So this is Hercules again. This is the main collection ROV that's used on the Nautilus, and it has explored to great depths. It has great endurance. They've looked at ancient shipwrecks. They've explored hydrothermal vents. They've found species new to science. And just for a size comparison, it's about the size of a Jeep. So the ROVs are central to the mission of the Nautilus and the exploration. Hercules can dive to 4,000 meters or 13,000 feet. And on our first dive, that was the deepest Hercules had ever gone. They did get it down to 4,000 meters. It can carry 250 pounds of samples. And believe me, the scientists wanted it to carry more. They were constantly deciding, do I keep this rock or that? I like that rock better. No, I like this rock better. So if you look at the picture on the left, you can see there are all kinds of gadgets on this ROV. Like I said, it's basically a diving robot. And a master pilot controls these operations below the ocean. Our master ROV pilot was this one you see in the middle, Jessica Sandoval. She was incredibly smart, doing amazing things. She was recently honored by the Explorers Club as one of the 50 people changing the world through exploration. She's a big deal. I met a lot of people who are a really big deal. This picture, she took time to show me how she controls the ROV. This is basically a little robot arm she holds called Master, and she moves it around, and her movements there are mimicked by the robot arms on the ROV. So samples are collected with that arm manipulator that you can see in the picture. And also they have something they call a slurp sampler, which you can kind of guess what it does based on that name. It's a little vacuum hose. Here it is vacuuming up a sea pig, which is a type of sea cucumber we collected one day. And then all the samples end up in this contraption called the BioBox. So it's kind of a sliding door, slides in and out of the ROV. The arms can put something in it, and then it closes up to bring those eventually to the surface. And you can also see these little things are called push core sediment samples. So they can push these into the surface and collect samples to see what is on the bottom of the ocean floor. They can collect water samples, they can collect all kinds of things, and they bring those back to the surface. And all of their information is available to scientists all over the world. Most of the dives are 24 hours long, and the longest that Herc has been down was 72 hours. The deepest dive, like I said, to date is the one that I was actually on. So the primary responsibility of the ROV Argus, the other ROV, is to keep an eye on Hercules. Argus from mythology was the seeing eye. And so they are tethered together. Hercules is below, Argus is above, and they use cameras on Argus to watch and navigate where they're moving Hercules. So the pilots are using the video imagery to know how to fly Hercules through the water. It's just an incredible system. And for size comparison, this is my little Chesty lion who traveled with me on the ship. We took lots of pictures with Chesty. And Argus is about the size of a minivan. And in theory, Argus can actually dive to 6,000 meters, but it just doesn't have all the same capabilities, so it's not the main ROV that's used. And in this picture in the center, you can see that on the ship itself, kind of where these ROVs are located and how they are deployed into the ocean. This is a crane that Hercules is attached to, and a crane operator will sit here and swing it over the edge and lower it into the ocean. Again, it's tethered to Argus, and Argus basically just gets pushed off the back end of the ship. Poor Argus. Doesn't get any of the glamour. All right, so the way all this works is something that OET has designed that they call telepresence. So we're out in the middle of the ocean, of course, away from Wi-Fi and internet, but they have established a system to send this information around the world in real time. So video and data streams from Hercules and Argus go up through fiber optic cables that are connected in the control van, which is where this picture is taken. That's where we would sit when we were on dive watch. And then it's sent by satellite to a receiving station and then onto an area called the Interspace Center in the University of Rhode Island. And from our control hub, video and data are distributed directly to scientists' computers all over the world and live to the World Wide Web. Telepresence frees the scientists from the constraints of being on the ship, being in remote locations, and it also allows scientists to collaborate in real time around the world. And that part was really exciting. I'll talk a little bit more about that in a minute. So, as I mentioned, telepresence allows us to live stream the dives directly through the internet. And this is the homepage of the Nautilus, where the live streams are being produced. So visitors tune in from all over the world to see exploration in real time. And that's what's so cool. People around the world are seeing things maybe for the first time at the same time as the explorers and the scientists. It just gave me chills when I was sitting there being a part of that. And this was where my job fit in on the ship. I was a science communication fellow, so my primary responsibility was I was a part of a dive watch team, and my job was to help the world audience understand in layman's terms what was happening, what we were seeing. And I also facilitated live conversation with the other members of our crew, the scientists, the ROV pilots, and the navigators on watch. We call this the Science Party Line, and it's, again, the live broadcast of the dive watch from the control van. This was a very special line because Dr. Geller came and sat next to me. I was a little nervous, but you can kind of see how we were distributed in the control van in the back row. The science communication fellow was always in the back, and then in the middle is our lead scientist. He was a geologist who studied underwater volcanoes, and then the person who collected all the data was in the back and kept records. We called him the data logger. And in the front row, we had the navigator on the far right. In the middle, we had the two ROV pilots. And then to the left side, you can't see in this picture, are the videographers. And I will just tell you that the video portion of this was mind-blowing. The live technology that they had to master. And I met amazing people, even from the video crew. They were people who worked on the Olympics, people who have already made a big name in nature, photography, and videography. It was just incredible to meet these people. So our crew would work 4 hours on, 8 hours off. So my dive watch was 8 to 12 in the morning and 8 to 12 at night. My second job was to teach classrooms about deep sea exploration all over the world. And so that was a really fun part for me. And because of the time difference, and people all over the world teaching live, I taught at some very interesting times. My earliest class was taught at 3:50 in the morning, and my latest class was 11 o'clock at night. So I didn't get a whole lot of sleep while we were in the diving portion of the journey. I taught as many as 5 classes per day. It was a little hard to juggle the watch shift with the teaching, but I love the teaching so much because that's what I was born to do. And I really enjoyed especially interacting with international students. My 2 favorites were this class from South Korea and a class in the Czech Republic. It was just so interesting to see how their classrooms were different from ours and similar, and how excited the kids were. I also got to teach all of the biology students at Lawrence High School in real time in one day. It's a lot of work. This picture was actually taken in my neighbor at LHS in her classroom, and that's me on the big screen teaching her students. My third job was getting to help in the wet lab, so actually doing science, which was really exciting for me. You can see on the left side, we are preparing a specimen that came up with the ROV, and they were prepared in either ethanol or they were frozen. And these specimens are sent to institutions all over the world, but the data is available to anyone. They don't have any restrictions on using this data. The woman on the right is a PhD student that I worked with. Her name is Coralie, which I thought was awesome for someone who studies the ocean. But she is filtering water samples in this picture and looking for traces of cobalt. So cobalt's a rare and valuable metal used in lithium batteries. So you can imagine the demand for materials such as this. And undersea mining may be our future. We may be extracting these minerals from the bottom of the ocean. It's only mined in a few places on the surface of the planet. And that's one thing I want you to remember. We're thinking about starting this whole new technology, but we really don't even know what impact that would have because we don't understand what's down there. These are some other samples from the wet lab. One thing that was cool about the wet lab is that, again, we have cameras all over the ship, and if you're logging on to the internet and you're looking at the website, you can look at different views, and Mike was able to watch me working in the wet lab. He had the cats watching too, and captive audience. And it's just so neat to see real-world science happening in exploration. Parts of the planet have never been seen before, and all these viewers are allowed to come and watch as well. Many of them are very dedicated, like regulars who log on every night. So it's really fun kind of getting to know their personalities too. So these are some samples that we brought up. This is called bamboo coral on the left side. In the middle, you see some of these iron manganese nodules. It takes 1 million years for 1 millimeter of this to deposit. So very slow deposit, very old rocks. On the right side top is a glass sponge. Most of the sponges we're familiar with. are not glass sponges. These are quite beautiful. They have silica in them. On the bottom we have a ravioli sea star. You can see where that name came from. And a sea urchin and some sea lilies. Another thing that was happening in the wet lab is eDNA extraction. eDNA is a whole new field that's so exciting. So one thing that we would do is we would not necessarily take an entire sponge or coral because we don't want to destroy those habitats in that ecosystem and take whole live organisms. So they often would take the robot arm and just snip a little sample. So they'd snip a little fragment and bring it back, and they can analyze the DNA. But they can also just take water samples, and fragments of DNA that are left behind can let us know who was there, even without making a direct collection. It would be like if you all left the room and we were able to take an air sample and figure out who all was in here just based on that sample. So it's a really cool, non-invasive technique of understanding what's in the environment, what's been there. And here's little Chesty sitting on one of the rocks that we collected. We collected 1,500 pounds of rocks on this expedition. I was there the day the FedEx guy came to the port. He was like, what? He said, you know, this is going to cost a lot. We know. All right, and I wanted to show you some of the beautiful examples of life from the deep sea since they are so weird and not really familiar. So first, the sponges. So just as a little refresher, it's probably been a while since high school biology. Sponges are animals. They're simple animals. They're filter feeders. They don't have tissues or organs, so that makes them pretty unique. They're some of the most simple animals on our planet. So everything they do has to be through coordinated efforts through their cells. And most of the sponges that we saw are these glass sponges. This picture where she's wearing gloves and holding this sharp-looking object, that's called a spicule. So those silica pieces kind of act like an internal skeleton to keep the structure of the sponge in the form that it has. And so most of these sponges grow on stalks, which is kind of weird. But remember, they're feeding off of this marine snow and zooplankton. So the higher they can get, the higher they can get into the current, the more likely they are to have food to keep them happy and growing. So some of them were on stalks that were quite long, quite impressive. This one in the middle was one of my favorites. It's called the ET sponge. You can see why. And you know, scientists do like to have a good time sometimes, believe it or not. If you translate its scientific name literally, its scientific name means magnificent alien. I love that one. Here you can see on the picture, if you can see it, there are 2 green dots. And that's something, if you tune in and watch the live footage, you'll notice that over and over. Those are lasers that they use for measurements. So this is a standard distance, and it helps us to get measurements on the structures and the organisms we find. Here are a few more. Another one that looks kind of like a creepy baby on the left. Many of these sponges are quite delicate. They often, again, they have to capture their food inside of them, and then they digest it extracellularly, basically kind of spitting out enzymes, and breaking it down and then absorbing those nutrients. Many of the sponges have associates. Associates is a name that means they live together in symbiosis with another creature. They help each other. And many of these associates are not well understood, again, because we just don't really know what's down there. Every time the Nautilus goes and explores, they basically find a new species, new to science. So understanding the relationships is a whole other level. Here you can see a little shrimp living in association with this sponge. Some of them have these plates that grow over them, and kind of, as the sponge gets bigger, it sort of, the food kind of filters through it like a sieve, gets trapped inside. And so as these little shrimp move into a sponge, often they'll get trapped as the sponge grows inside of the sponge, and they end up spending their whole life there with their mate. And so it's very kind of strange custom, but a custom among the Japanese is to often give these dried sponges with the dead pair of shrimp inside of them as a wedding gift. Like, congratulations, you're trapped for life. You live until you die together. So there's some humor there. One more I wanted to mention of the sponges before we go on. This one in the top middle, it looks like a flower, is actually one example that it's not a glass sponge. It's more like the sponges that we're used to seeing, like if you go to Hobby Lobby and buy a real sponge for art. But it is a carnivorous sponge. So it's one of the few rare carnivorous sponges that traps the live prey itself. All right, deep sea corals. So again, the corals are very different than the bright, flashy ones that you think of, like on the Great Barrier Reef. Just as a reminder, corals are animals. They live in colonies. Many of them have a skeleton, some of them don't. And they feed by these little tiny polyps grabbing food out of the water. And once again, we often see associates. If you look closely, this thing is an echinoderm. I believe this is a snake star, kind of like a brittle star, one of the sea stars. And you often see over and over the same species of coral and the same associates. So once again, they don't always know if it's a mutualistic relationship where both are benefiting, or perhaps just that the sea star can get up higher and get closer to the nutrients. But they think there must be some beneficial relationships. And all these pictures, by the way, were taken by Hercules during our expedition. All right, look at the beautiful colors. Do you remember when I told you there's no light down here? So there's things don't see this beautiful color. So that's another one of the interesting things that we wonder about is, why are these colors down there? There's nothing to see them. There's also a lot of bioluminescence. So the colors give some of the scientists hope that some of these may be powerful antioxidant compounds that may hold medical cures. They've already found a deep sea sponge that they're using to treat pancreatic cancer. So these creatures can be very tall. Some of them can be 5 meters tall. They are very slow growing. We don't have a way of aging the sponges, but we do have some ways of aging the corals, and they know that they are sometimes hundreds of years old. So they're kind of sometimes these can be compared to the redwood forest, the deep sea ocean, the redwood forest of the deep sea ocean. Slow growing and very old. And here you can again see some of the associates. Here's sea star tangled up here again, and up there, and over here we have a cute little, let's see, that's called a squat lobster, the pink little guy that's living inside of a black coral. The black coral and some of these other deep sea corals are among what they call the precious corals, and they've historically, traditionally been collected for jewelry. So that's another threat to this deep sea ecosystem. I really love this spirally one in the bottom. It's called the fireworks coral, and they believe that this structure may help the nutrients to kind of funnel down into the organism, to get those to the organism. They're quite beautiful. Some other fun, weird creatures that we saw on our journey. These are things that I saw when I was on dive watch that I chose. This one is called the headless chicken monster, which is a pretty funny name, but it's also a pretty funny creature. They are a type of sea cucumber found in the deep sea, and it's actually very rare to find them sitting on the bottom of the ocean. They're typically seen moving through columns of water, swimming, and I looked it up, and apparently the video footage that we took is probably the longest that this creature's ever been documented sitting on the bottom of the ocean. It wasn't just sitting though, it was using its little polyps and shoving sand into its gut tract. Just shoving like a little sea pig. Just like the name. It was the cutest thing. You can see its guts. I mean, it's kind of cute and ugly at the same time. And on the right is a really special creature we found when you're not on dive watch, you're hanging around sleeping or working on a project or eating. And when we found this, several biologists burst into the cabin and wanted to weigh in on this creature because it was so special. We have cameras, video cameras all over the ship so people can see what is being viewed live in exploration. Some of the scientists just can't get enough of it. They watch pretty much all the time. So this creature is called a purple acorn worm. It also has a fun scientific name. Scientific name is Yoda purpurata. It's basically a purple Yoda. It was just discovered in 2012. It is from a group that are a sister group to the echinoderms, the sea stars, etc. And they share some traits in common with chordates, the group that we belong to, things with backbone and central nervous system. And this is the kind of link that we always want to study for evolution to understand the link between vertebrates and invertebrates. It can grow to be 1 foot long, and when we found it here, this was the first time this creature had been found in the Pacific Ocean. It was only known from the Atlantic. A few other creatures that have some strange associates. This one's kind of fun. The starfish is eating the coral, but it also has associates living on it, kind of little creatures living on it, so a parasite probably. And this pink sea anemone at the top, to me it looks a little bit like a muppet. And that one is probably just perching up high to get up into the nutrient stream. And then down at the bottom, a crinoid, once again, just hanging out up high, get up where the food is. Everybody wants to be where the food is. Probably my favorite thing that we got to see was a Dumbo octopus, and that was really special to me because one time when Dr. Ballard was a guest in my classroom, he came and did a Zoom with us. The kids asked him, "What's your favorite creature?" and he said, "Dumbo octopus." So I was really excited. We were the only dive watch to get to see one on our dive. They're quite hard to find. They're very solitary and rare. But this cute little guy was putting on quite a show for us. And if you go back to the website, they have galleries of video footage, and there's a whole video of the live footage of when we discovered this cute little guy. At the top is a beautiful purple sea cucumber, again, these crazy colors in areas where color isn't seen, and then a beautiful jellyfish of some type. It was really fun for me. I know that there were classes viewing live at LHS when we found that. I heard back from students about seeing that. And some fish. There aren't so many down deep like this. Many of the ones that we see have a body shape like the one on the left, kind of an eel shape. But probably everybody's favorite is this guy on the right side. He's called choncops. He is a type of anglerfish. Common name is toadfish. And he's just got that little resting pout face that everybody loves. And every picture of him is funny. It's so crazy to watch them on the bottom of the ocean. They move along like on their fins, kind of use them as modified appendages to kind of walk along the seafloor. And you can see a little bit of his bioluminescent angle or dangle here on the top picture. And then this poor guy on the bottom left is the laughingstock of the fish world pretty much, because his common name is the bony-eared assfish. Poor guy. Could have ended up with the worst name, and it's notorious for having the smallest brain compared to the body size, pretty much in the vertebrate world. Poor guy. Evening entertainment was shark watching by sunset. Always the sunsets. This is an oceanic rights of shark. They often came and swam around the boat. And then if you had any free time and didn't need to sleep and you weren't seasick, there was always a gym available for you. Of course, you shared the gym with the negative 80-degree freezer and portholes and a moon pole. The moon pole is a hole that goes down to the bottom of the ship for connections, wires, etc., pipes. And I tried it one day, and you get an ab workout even if you're not moving because you're just trying to stay still. So, woo, it's not for everybody. My final thoughts. We still have so much to learn about the planet that we live on, and more people have been to outer space than to the deep sea. And really the important thing is we can't protect what we don't understand. These ancient, slow-growing deep-sea coral and sponge gardens can be many hundreds of years old. They're slow-growing. They're threatened by pollution, deep-sea fishing, dredging, coral collecting, and mining. The state of Washington has already proactively banned deep-sea mining on its coastal waters, which is interesting. These areas act as nurseries for things like sharks and other living creatures. They host these complex relationships that we're just beginning to understand, and then they hold medical cures, and of course, great biodiversity. So we need to understand what we're doing before we allow any of this development to take place. And also, it was impressed upon me how collaborative science has to be. All these scientists working in real time together. And it was just a delight. There was a whole separate back channel for just scientists that I could see the text, so if they found something And, my lead scientist was a geologist, so he wasn't an expert in understanding these things. So he would log on and say, like, hey, so-and-so, do you know what we're looking at? And it was just so incredible reading the emotion sometimes of scientists who were seeing things for the first time, or maybe the second time in their life. I've only read about this, I can't believe we're seeing this. It was just awesome. And even with my limited knowledge as a teacher, I was definitely no expert. Sometimes I was able to help them with identification just because my colleagues who were also SCFs on their dive watch, sometimes I was going through editing their photos, and I would say, hey, what's that? And they would tell me, and then on my dive watch one night we saw something and no one knew what it was. I said, I know what it is! Just because I had been working on the photos and the videos. And then the last one, the most important one, I think for all of us, is don't be afraid to be a dreamer, no matter how old you are or how young you are. This is a quote that I wrote down the first time Dr. Ballard came to talk to us at LHS. He said, never climb a small mountain, pick a big one, because there are very few people with the courage to try the big ones. And I never would have dreamed, really, as a kid, that when I read my first science book that made me want to be a scientist, which was about the ocean, that I would actually get to travel the ocean and teach about the ocean and experience deep sea exploration, but it happened. And if you would like to be more involved with ocean exploration, there are a couple of great opportunities. One is they are off season right now. Their new season will start again in the spring. But if you check out their website, they offer live ship-to-shore interactions typically between about February or March all the way through October, but our year was as far as into December. And so these aren't just for classrooms. They could be groups like this, or like a Bible study group, or just a group of friends, and you can apply to have these talks done live from the ship by the SCFs. And then anyone can follow along on the website. And again, you yourself can type in questions. That was my job as an SCF, is to say, hey, so-and-so from Australia wants to know this. But they also turned into a neat cultural connection as well. We had worked there close to Christmas, so we ended up chatting late at night with people in New Zealand and Australia about the specialty breads that they made for Christmas, and asking them questions on our long dive nights when we had nothing else to do. And it was just so neat to learn about people around the world. Again, we're so different, but we also have so much in common. So check out their website, and when the live season starts again, you can also be part of this exciting exploration firsthand. So thank you very much, and I'm glad to answer your questions.
About your experience with plastic in the ocean, whatever, maybe you said something.
Yeah, no, that's a great question. Plastic and pollution. Since we were so deep, I was quite surprised at how little trash we did find. But probably the most interesting thing we found was a single flip-flop. A single flip-flop sandal. We would occasionally find and pick up things like old beer cans. Some of them were quite, like 30 years old. It was kind of a guessing game. What year is this from? The best story I heard was that one of the most interesting pieces of trash that another crew member found was a rice cooker. And they went down and they were going to pick it up to carry it back to the ship. And they opened it, and an octopus popped out, and it grabbed it, and it shut it again. So they left it, and 2 years later, they went back to the same place, and it was still there, using it as a home. Any other questions?
I thought, as you were talking about what was used in pancreatic cancer, and I thought, oh my gosh, we start finding great uses for these that will make money. What kind of control, regulations do you see, and who's going to do this?
Yeah, that's a good question. It depends on where you are. We were mostly in open ocean, but part of the time we were in Honolulu National Marine Sanctuary. And if you're in an area that's protected like that, there are a lot of guidelines for how you can move through that area or collect. But more in the open ocean, there aren't a lot of rules, and that's probably one of the concerning things is once you're beyond those boundaries of where countries claim the land, there's not a lot of regulating going on. Some of these deep sea corals are now protected, thankfully. That doesn't mean they're not being harvested, that they are protected. So.
I don't know if you have enough base material to answer this yet, but can you comment on the impact of human society on the biodiversity where you were?
That's a good question. Human impact on biodiversity in this area, and I think that is really impossible to answer because we're just now surveying biodiversity, so we can't really know what was here. I did ask the scientists about the impact of climate change at these steps, and they were somewhat hopeful that perhaps this is so far down that maybe our climate change disaster hasn't really made much of an impact there yet. But we do know that some areas have been decimated by dredging, deep sea dredging, dragging nets and things along the bottom of the ocean. And of course pollution, there's no boundaries in the water for pollution, chemical pollution. However, I'd like to think that, since we haven't had the technology to explore this stuff, perhaps we're at that great stage where we can protect what is there now before things get more developed. Any other questions?
The effect of seeing bioluminescence attracts my attention. I'd like to know how far down can you see bioluminescence? Do you have any idea of the sources of molecular species that are used in detecting bioluminescence? Because you of course, don't have light down there. Does it go on forever, or are there limitations? And has anybody ever thought of taking bioluminescence as a lighting material to show up what is going down in those darker regions?
These are great questions I can't answer much of, but I'll tell you what I know. Definitely not my field of expertise. They said that most of these creatures have some capability of bioluminescence. And I asked why, and one of the examples they gave us is that if a coral is being attacked by a sea star and it gives off this flare of bioluminescence, perhaps a fish will come along and be attracted to the sea star and eat it. So it might be a defense mechanism for them. But I don't know enough about the chemistry there to know, it's probably signals, mating signals, finding each other, defense, but I don't know enough about the chemistry behind it to understand, but yeah, there's great promise there. Yes.
I wondered, you said you saw a flip-flop, that's the size of a foot. Did you scan for microplastics? Did you take water samples?
They're collecting water samples all the time where they can do that, so they have a whole library of water samples where they can detect that. And they're also collecting lots of microbial information, all kinds of microorganisms living down there. The expedition before me was really focused on microorganisms. There's a lot there that we couldn't even see that was being collected.
I was going to ask you, you made one reference to seasickness. How did you accomplish so much on the Pacific?
Yeah, great
know what
question.
it's like.
Yeah, especially during that season, I found out later that people were like, "Oh, you're going around the holidays." I will just say that I was not prepared for the amount of movement on the boat. I was kind of thinking, "Oh, no big deal." The only ship I'd been on was a cruise ship in the Galapagos. It was smooth sailing, piece of cake. And Mike was laughing at me when I took the assignment, like, "You're going to be seasick the whole time." I'm proactive about these things, so I got a patch for seasickness from my doctor. And it was kind of funny because the crew members were kind of laughing at me, like, "Whoa, that's a little bit much to go ahead." But at the end, some of them were asking me about the patch. Because I had one 30-minute period where I felt really queasy. I was fine the rest of the time. But it was so, just to give you an idea of the movement, when we were actually out moving, especially mapping, because we were moving along at a really fast clip when we were mapping, if you wanted to move across the room, you kind of have to sit there and think about it, like, how am I going to get there? And then you grab onto everything, there are handrails everywhere. It was a lot more movement than I ever thought there would be, but I was really lucky. All right. No more questions. Thank you very much. If you have to talk to any of you, have any individual questions, again, thank you so much for being here. I've enjoyed it so much.